Greetings, this is your vodcast for Wednesday, March the 12th, and the third of the four for the period of time that I'm gone in New York. We will have gotten through 2 races at the end of today, and hopefully are kicking butt. You guys are probably also starting to get pretty darn stoked because spring break is beginning to become near, and I'm hoping that most of your spring break you will spend away from microbiology, that you'll spend some time on an adventure, maybe headed to Moab or somewhere else where you can get some recuperation and rest. But I also hope that you'll spend a little bit of time with your epic metabolism and gene expression poster. Remember that that poster can contain all of the class content as long as it is in a pictorial form of your own making and it doesn't have large blocks of text on it. It needs to be on a single sheet, but otherwise it can have absolutely everything that we have covered. It just needs to be situated within a cell and be looking like what happens within a cell. I'm also hoping that over spring break you might pick up 1 or maybe 2 articles that might relate to your group poster presentation to allow you to keep perpetual motion on that project. Perpetual motion is probably the #1 key for getting large projects done. And let me tell you, I know how hard it can be to maintain that, but if you do, it certainly makes a project into something much easier. Lab, we've got a few up-and-coming things, and one that I had reminded you of last time is that your skill test rubrics are going to be due to your TA on the first day of lab after spring break. That enables you to use this entire week to finish up your skills test. That means, though, also that you only have 1 attempt on each one of those skill categories. So make sure you get those finished up and ready to turn in the week after spring break. The other exciting thing that is happening tomorrow is that you get to become detectives and to do some diagnostic bacteriology. That is, you're going to receive some unknown bacteria, you're going to get clues along with those unknowns, and you're going to be able to make hypotheses as to the identification of those particular unknown bacteria. However, we want you to submit not only a hypothesis as to the identity, but we also want you to submit a plan for how you're going to go about identifying those bacteria. So over the last couple of labs, you've been learning about how you can do biochemical tests to give you a profile on an organism and allow you to know what that organism might be. So you're going to predict for your hypothesized unknowns, you're going to give a flow chart or a verbal description of the particular biochemical tests that will be important for you to perform in order to identify your unknown. So this is basically a plan for identification that you're going to submit along with your hypotheses and references. So that being said, let's hop back into the epic translation journey. We had been talking about the initiation phase of translation, and we had talked about the assembly of the ribosome at that initiation site. We had mentioned that the N-formal methionine carrying tRNA was bound and was getting ready to begin the process of translation. At the very last moment, the 50S ribosome came on in and added to the complex. And at that point, the whole complex was ready to head from the initiation phase into the elongation phase. And that's where we pick up the story today, is with elongation. We have our 50 and our 30S ribosomal RNA subunits, and they are currently bound to the mRNA. Remember that there is the mRNA binding site within the 30S subunit that is that pyrimidine-rich sequence that binds with the purine-rich Shine-Delgarno sequence. So we know that that had already happened. Now, we need to define within the ribosome particular sites that at which different events are going to occur throughout the process of elongation. These sites are pretty easy to remember because they are termed the E, the P, and the A sites. So think EPA, and you have your sites of the ribosome. The P site stands for peptidyl. The A site stands for aminyl acyl or acceptor site, and the E site stands for exit. And you would guess right there is where tRNAs make their exit out of the ribosome. To reorient ourselves where we left off last time, let's just remind that we had the mRNA that was passing through right here, being ready to be read by the ribosome. And after initiation had taken place, the only reading and adapting that was currently being done was that in the P site, the tRNA, and this is going to be a horrible tRNA drawing, just fair warning, the tRNA that was carrying on its acceptor stem N-formal methionine, or FMET, so this being FMET, was currently bound at the P site. So at this moment, just after initiation has finished, there is a filled P site with the N-formal methionine amino acid in it, and an open A site. And that is the site, the open A site is the site where the next tRNA carrying the amino acid that will correspond to codon number 2 is now going to bind to the ribosome. That's where we'll pick up elongation in our next image and show the open A site ready for binding. So we'll see now that the A site will soon be filled by the tRNA carrying the second amino acid. Remember, of course, that this codon is going to be what specifies what amino acid is on that acceptor stem. So as we know, although I'm showing these as different colors, what they actually represent are different codons. And we know that the first codon is AUG. If the second codon is CCA, for example, and let me use a better color, a more visible color to write that down there. If the second codon is CCA, you all can go ahead and use your codon usage table to look up what amino acid is carried on the acceptor stem of that particular tRNA. And, or we could say that another way, what is the second amino acid in what will be a growing polypeptide chain as elongation begins to ensue. So what we can say is that the process of elongation has 3 phases of its own. Those 3 phases are tRNA binding, and that's when an aminoacyl tRNA is the technical term for it. tRNA binding. The reason that it's called an aminoacyl tRNA is because it is bound to its amino acid. So the aminoacyl tRNA binds. Number 2, so this is 1. Number 2 is what we call transpeptidation, or said another way, formation of the peptide bond. Transpeptidation is just the sexy term for it. Number 3 then is translocation. Translocation literally means that the ribosome moves on down. So it moves such that suddenly what is in the P site goes to the E site, what is in the A site goes to the P site. But in our image right here, the first thing that has to take place is transpeptidation. So the next step that we're expecting is for a peptide bond to form between the amino acid in the P site and the amino acid in the A site. We know that the catalyst for that peptide bond formation is the 23S rRNA that lives in the large ribosomal subunit. That is, it acts as a ribozyme. So let's look for transpeptidation being our next step. The covalent bond, the peptide bond, formed between the N-formal methionine and the second amino acid in the chain. Now we're ready for translocation. The ribosome literally is going to take a step, a single step, 1 codon down the mRNA transcript. It's going to go such that whatever codon is in the P site will slip into the E site. That means that this tRNA is going to leave behind its N-formal methionine amino acid, and this tRNA is going to make its exit. It's going to head off so that it can activate another amino acid and play another role as an adapter. So what's cool about this is as the elongation phase continues, the ever elongating polypeptide chain will make its way out of the top of the 50S ribosomal subunit through an actual pore in the subunit. That is an actual opening. And we'll see a longer and longer polypeptide chain with each step of elongation. Notice that the amino terminus will be the first thing to come out of that hole in the top of the 50S subunit. So in translocation, there is a movement down and the tRNA leaves through the E site. We'll see that happening. So now what we recognize is that the P site currently has both the amino acid from the initiator tRNA and the second amino acid that was bound to the second amino acyl tRNA. This leaves open the A site for the binding of yet a third amino acyl tRNA, which has its amino acid, whatever corresponds to the third codon in the sequence. So this process keeps on happening, and transpeptidation will happen next, followed by translocation, until finally we get a very, very long polypeptide chain. Well, of course, the length depends upon how long that eventual protein is, how many amino acids are found in whatever protein product is being made. So the same mRNA, interestingly enough, can be translated at the same time by different ribosomes. That is to say, the minute that the Shine-Delgarno sequence and the AUG start codon are exposed, we can see the initiation complex form again, and a second ribosome begin to read the same mRNA at the same time that the first ribosome is reading it. This leads to the formation of something whose name you could probably guess. That is, it is a polyribosome, or for short, people just like to call it a polysome. I would love to have a model of a polysome. So if anybody wants to get jazzed on a little bit of extra credit, remember you max out at 15 points, but definitely make me a model of a polyribosome. You could even make it so that at least one of the ribosomes actually shows the EP and A site, and that would be a really, really neat model. If you did a good job of it, I would definitely show it on future cheesy videos, no question. As you would guess, the next step is termination. So when it all comes to an end, the thing that causes that is when the ribosome reaches a stop codon. So as it moves into a position where what is currently found in the A site is a codon or nonsense codon, that is one name for it, meaning that it doesn't encode for any amino acid. At that point, this leads to termination, and it causes the ribosome to fall off of the transcript, and we get the dissociation of the polypeptide chain, and it's now ready to fold and or be further processed. So that is what's going to be next. But notice that we have our amino terminus exposed out here, and recognize that this process will terminate whenever a stop codon or nonsense codon is found there. So at this point, proteins called release factors assist in this process, and the ribosome falls off of the mRNA and dissociates again into its 30S and 50S subunits. The 30S subunit can go back and begin another round of translation, binding again to the Shine-Delgarno sequence, aligning again with the AUG start codon. So this process, as it comes to an end, can also begin again, if you will, and an mRNA transcript can be transcribed many times until finally, of course, it will be degraded. So the lifetime of an RNA is not forever. Eventually, that RNA will no longer be able to be effectively transcribed. It has a limited lifespan. And that's why, hey, that's why DNA and not RNA is the primary genetic material. So after translation has terminated, we get post-translational modifications. And one of those modifications is something that you're going to be very I guess, very familiar with, and that is that a signal sequence may be added on to the protein, the amino acid sequence. And that signal sequence, remember, its role was so very important in sec-dependent secretion. So if there is the addition of those few amino acids at the amino terminus, then that signals that that particular polypeptide chain should be taken to the sec apparatus, where sec B will bind it, keep it from folding, and the sec YG and sec A will play a role in getting it out of the cell. So remember how familiar we are already with that process. Another thing that can happen post-translationally is that folding may be made possible by something called a chaperone. Chaperones are large proteins that literally allow for self-assembly of other proteins. They have in them a cavity within which other proteins can be coddled and allowed to fold without outside influences. It's like a chaperone creates a safe haven for the protein in which to fold. The other thing that happens post-translationally is we actually get the removal of the first of the amino termini amino acids. So about 1 to 3 amino acids on the amino terminus are always removed, and that always includes the F-met. So when we have the sequence of the polypeptide chain, for example, of the polypeptide chain that we just looked at, the N-formal methionine would most likely be cleaved off. So at the most amino terminus, we see that we have the binding of that formal group, onto the most amino terminus, and the R, and the second amino acid. And hopefully some of you looked that up and you found out what the R group on that would be. So this would be a methionine R group. And if you looked up the amino acid that I just randomly made up, that it was CCA from the codon usage table, then you found out that in fact the R group here was the very kinky proline. So that was our second amino acid. And one of the notes that we could make about this is that most likely in post-translational modification, both of the first 2 amino acids that we talked about, would actually be clipped off. So generally you don't see an amino acid sequence beginning with N-formal methionine, because usually in post-translational modification, that N-formal methionine is actually lost. So when we see an amino acid sequence, maybe it's something like, V, V, L, L, P, and you're looking at that and you're thinking, shouldn't it start with N-formal methionine? Well, it probably did, but in post-translational modification, it got cleaved off. So I'm sad to say that I'm not actually wearing my ribosome t-shirt today, but I do have it with me, and so I'm going to bring it back up for you to take another good look at. It's very nicely done. Some of you will appreciate the artisanship here, and I want to show you the full glory of the t-shirt and show you particularly what's going on within the ribosomal subunits, and to recognize the EP and the A site. And she's put EP and A site on there, really nicely done. You got the mRNA being read in the yarn there, the awesomely colored yarn. You've got the nice elongating polypeptide chain, and even note that she made the initial amino acid N-formal methionine. But there is a mistake in this t-shirt. So you're noticing right there perhaps what might be the mistake. So she's got the tRNA bound at the P site there. And so I have decided that whomever emails me fastest after watching this vodcast is going to, with the mistake, what is her mistake, is going to actually be the happy recipient of Rex. So send me your emails quick.